So I'm going to talk about identifying regulatory elements in the UTR for also potential therapeutic targets, but also just what insights can we get from this? Okay, so the basic questions for developing therapies are the what, the where, the how, and also the when in specific cases. And really today, they're all very interconnected. In general, today we will focus mostly on addressing some of the what and some of the where. And I'll start by describing the first project. I'll spend most of the time there. And I'll just say if there are people in the audience that attended a recent meeting in Switzerland, where I also met Alicia and Julian, etc., then I apologize. Most of this is based on that talk with just like one or two slides maybe added recently. Anyway, moving on. So the first project I'm going to talk about is an RNA language model for identifying G-quadruplexes and disease variants from population scale genetic data. So the first question you might be asking yourself is, well, what are those G-quads? So G-quads in RNA are basically a secondary structure. You can describe it either with a sequence motif as shown here on the left, which are G-runs with spacers in between. And you can also describe it with the cartoon of the structure that they form here on the right. But the thing is that this description that I just showed you is pretty crude, and we know that. So we know sequences that not necessarily conform to this specified motif on the left actually would form structures. Sequences that do conform to this do not, right? And we also know that the representation on the right for structure is also very crude because we know there are different variations to these structures. These are called like as subtypes of G-quads, as some of them are illustrated here below. So we definitely want to have, and I hope to convince you later, we want to have a better refined definition of the motifs and their structure. So where can we find them? A high-throughput assay was used to map them in mRNAs, and they were found mostly to be in the 5' and 3' UTRs, although there's some in the coding sequences as well. And we know through numerous studies that they can affect different RNA processes, like the rate of translation, alternative polydentylation, and alternative splicing. Now, the typical way, or the sort of classical way of identifying them is with an experiment called circular dichroism. Okay, this is based on shining wavelength, different wavelength, and watching the absorption. And you get this typical curve that is shown here, which would be typical for forming the structure. Another way of measuring it, which is also commonly used, is using potassium ions as shown here, or lithium ions, which are weaker. The potassium stabilizes the structure, and then what happens is when you do sequencing, during the reverse transcription, it gets stalled. So when you do the sequencing, because the reverse transcription got stuck here, your sequencing reads are going to be stuck here and going to end here. So you can compare with or without the potassium and see the differences where reads tend to get stuck. And by this, you can get a readout of where those structures have formed. Okay? And this was done first time in a high throughput manner and led to this Nature Methods paper back in 2016, reporting their widespread of those across the RNAs. Now, almost at the same time that this was published, another paper was published in this obscure journal called Science by another unknown group, David Bartel's group, basically saying that those don't actually form in vivo, right? So do we have these very opposing view of RNA G-quadruplexes, which started what we now call as the G-quad wars that raged on for several years and have known to be affecting many PIs, research addendas, and grants. Now, we came to this area much later in 2020, And we published this paper, which is, of course, a great, beautiful paper, which I recommend you to read. But the main point here is that instead of focusing on how those structures are forming, if they're forming, we took a population genetics approach and asked whether we can find negative selection that shows that they are actually function and what other things that we can find, like RNA binding proteins and etc. And I'll just highlight two of the main conclusions that were derived in this paper. One is they're very clearly in the UTR under negative selection. And the other one is that disrupting them is associated with changes in expression. Okay? But there are also very clear limitations to the study. The way we did this, we only looked at canonical G4s and focused only on the ones that were validated with this RT-stop method that I mentioned, which was not only in HeLa cells. Okay. So we wanted to extend this and answer basic questions regarding RNA G-Quads. We wanted to know, are they really functional? Are they associated with any specific disease? Do they affect protein expression in vivo? Right, so that's important. And do variants affect the expression or the structure? Okay. And to go about answering the questions, we sort of came up with the following pipeline. We start with the available high throughput data, and then based on that, we train a model. Once we have the model, we can run predictions and we can analyze associations with phenotypes for RG4 deleterious variants, and then we can also do validation to see that that's indeed the case. And that's basically what we went out set to do. Okay, so let me now describe what we did. We first built a mRNA language model over all mRNAs. This is a BERT transformer. Details are less relevant now for the specific talk. But once we have this, then we fine-tune it on both negative and positive labels from the available data. So we have about 10,000 of those, 5 and 5. And we train it to do two tasks. One is to predict formation of RNA G-Quads, and the other one is to predict subtypes. Okay? Now, so these are the main tasks. And how well did we do? We evaluated extensively against other algorithms. This is the latest and greatest that was published before. This is a CNN-based model, and we can see some improvements in just predicting whether an RG4 is there, yes or no, based on this specific data set on the high throughput RT-stop. But when we're trying to predict subtypes, there is a big difference, potentially because the CNN is not as good as identifying those connection or structural elements. So that was one. And of course, then you want to see that you're able to generalize to other data sets, other experimental techniques, etc. So we used the G4RNA that was published by Michel Scott and colleagues, Jean-Pierre, from Sherbrooke. And these are what you see here at the bottom are 5 different experimental techniques. And this is the amount of sequences for each one of them. These are the 5 most common ones in this database. And then for each one of them, we broke down performance and compared ourselves to 3 other algorithms, some of them based on motif searches and different versions. And another one is a CNN. And this is, again, a transformer language model. And we can definitely see differences. So you can see here, circular dichroism that I mentioned before shows here on the left, and everybody seems to be performing pretty well there. But the one in the center, which is a luciferase assay, there are big differences. And then when we looked into what may cause these differences, we noticed that there is a very different, big difference in the length of the sequences that were evaluated. So this graph shows the same data, but now the x-axis breaks it down by the length of the sequence that's being evaluated. And the sequences on the left are very short, and they are highly enriched for circular dichroism. And you can see that basically all algorithms do pretty well there. But as sequences become long, the differences become much wider. Okay. And again, we are the red diamonds here. Um, and that made us think because what happens is for decades, when people want to validate RG4s, they go to circular dichroism and they like to use short sequences for experiments and testing and testing different elements of this. But the problem is, it seems that with those, it's a relatively easy task and everybody seems to perform well. It's unclear that this is exactly what's happening in nature necessarily. So we went and looked at that. And what we saw is a very big shift, right? So although people like to test short sequences, the distribution of length shown here as a CDF for circular dichroism experiments that have been collected through decades of research are very much shifted to the left. They tend to be significantly shorter than the ones that are identified by RD-stop, and even more so the one that we find naturally occurring for canonical RG4s in the 5' UTR. So basically, it tells us, yes, there is a difference in performance between the algorithms, but also it seems to be that we've been focusing on things that are short because maybe it's easier to measure, and then we introduce a bias. So that means every algorithm that then we develop would tend to focus on those or will be biased in its evaluation. So that's maybe an important point to consider in other domains as well. And once we have this algorithm, of course, we can evaluate different things with this G-former model. So when we look at breaking versus non-breaking, whether it's within the G-run or even not within the G-run, we see significant difference, again, in allele frequency, again, indicating negative selection. And when we look at short versus long, coming back to that question, again, we see a difference. So there seems to be more negative selection in terms of frequency of allele frequencies on the longer ones, actually. So indicating they might be functional or maybe more sensitive to this as well. We then look at the distribution of the various subtypes. So this is the distribution that we had in our training data from RT-stop, and you can see a high representation for long loop, canonical, and bulges. But when we look at the transcriptome and the RG4 breaking ones that we found in the transcriptome, RG4 breaking by the predictions of the algorithm, we see a very significantly different distribution. We see much higher enrichments for those motifs with high G content around them, and we see way less canonical ones. Okay, and we see this for the 5' and we see the same thing for 3'. When we look across those subtypes for the effect of genetic variants that break them, again, we see a significant difference between the subtypes. Is shown here both for 5' and 3'. And that makes sense if we think of what we know about those subtypes in terms of their strength, and that also aligns well with recent data. So these are results from a recent bioarchive from Dan Dominguez's lab, which conveniently came out recently with high throughput measurements. And what you're seeing here are at the x-axis the length of the loop, and the dashed line is when they call it as forming a structure or not forming a structure. And we can definitely see whether you have a GGG track or a 4Gs track, the trend stays the same, where as you increase the loop length, the strength of the structure drops. So that's very much in line with what we see as well. Okay. What about nucleotide composition outside the G4s? We find that they have significant effect. For that, we run EIG, enhanced integrated gradients. That's a method to interpret deep learning models that we published back in 2020. So we apply it here, and we see a very strong preference to Gs. Again, think of what I showed you before about those G enrichments, but also for potentially As and some preferences not to have Cs. Again, that would make a lot of sense because if you have those Cs, they're going to try and bind with the Gs for strong competitive structures. So that makes a lot of sense intuitively, but the point I want to make about this is these are just marginal distributions per position. Zero is here is basically the edges to upstream or downstream of the G track that we're looking at. But these are marginal distributions per positions. When we did this for dinucleotide or trinucleotide, you see that context really matters. So you can tolerate Cs in specific situations. So really, it's very much context specific, which is not captured by this specific plot. And again, this is very much in line, this importance of those flanking regions learned by the model is very much in line with high throughput data from that bioRxiv preprint. And this, what you see here on the x-axis, are 6 different wild-type G-quads that are known to form G-quads, and they introduced thousands of mutations around all of them. And you can see that some of them increase and some of them decrease the score. And again, the dashed line is sort of the wild-type baseline for forming G-quads. And here are 2 examples of 2 of them, right? You can see the regions where the G tracks are, and positive and negatives here are measured as red to blue variations. What I want to point out is you can see a lot of those red and blues. Yes, you see, of course, if you break the G track, of course, that's bad. That's sort of what everybody knows, but they don't value them the same. And you have a lot of importance for those flanking regions. Some of them can decrease or increase the formation. Again, makes it very much context dependent. We also looked whether those regions can have are enriched for specific RNA binding protein motifs, because we know some of them like to interact with this, and we do find high enrichment for some of them. Specifically, we find a lot of GA motifs like HNRPF or H, which are known to bind around G-quads, and there's been multiple publications around that, or SRSF4, or UA reach motifs as well. Okay. Now, if you can do all this, of course, what you probably wondering, at least, our reviewers definitely were wondering, and rightfully so, is how can you connect those in a larger scale to pathogenic variants? Can you? All right. And so here is what we got for enrichments in ClinVar for 5' and 3' UTR for disrupting G-quads, and we see a very strong statistical significance for enrichments of those for G-quad breaking variants. Now, of course, the p-value is high, is like 10 to the minus 10 or whatever. And it's interesting because if you look at the tail distribution, you say, okay, let me see a really big difference. Like you break it, like a change of at least 0.5, right? Of course, you get less, but what's the fold enrichment that you would find in ClinVar, say, for benign versus pathogenic, say, in the 5' UTR? Any guesses? Maria, can we do this interactive? Can they write their guesses?
Sure.
Because I can't see anything, but maybe you can say.
Yeah, please raise a hand or write in the chat.
So I'll give you the first answer for free. Okay, let's do it like that. So the enrichment we found is 5.2 fold. Okay, so you'd say, oh, 5.2, that sounds like a nice number. It's close to 5. What does it mean? So you're maybe wondering, how does it compare to other things? We were wondering as well. So we did the same trick for predicted variants that break splicing in 5' UTR. Right? Predicted by SpiceAI, which a lot of people use. What's the fold enrichment that we saw? Remember, for G-quads, we saw 5.2 fold. What did we find for 5' UTR SpiceAI disrupting variants? Okay, in ClinVar. Guesses?
In the chat, I see 6.5.
Sorry?
In the chat, I see 6.5. 1.1,
6.5. Okay, 6.5 going once. Anybody else?
1.1 also.
1.1. Wow, that person is pessimistic. Okay, what else do we have there?
15.2.
Sorry?
15.2.
15.2. Okay, what else?
10.
10. Okay, so the answer goes to the person who said the prize, which doesn't exist, would go to drum rolls, 15.2. Very close. Maybe they got our revised paper, so they read it. Okay, it was very close, and it was around 15, right? And you'd say, oh, so splicing is much more enriched. But let me just note here, if you think about it a little bit, there are two things to note, right? One of them as a mechanism, splicing can like really kill your UTR, right? You can splice out a whole segment. We're not talking about one regulatory element that you're maybe breaking. You can like take out an entire segment. So that's one thing to think about. And the other one is that ClinVar is highly, highly, highly biased, right? So people have been looking for things that would hit splice site and disrupt them for decades. They haven't been looking for G-quads being broken, right? So this set by itself is highly biased. So I would argue that 5.2-fold enrichment for something we haven't been looking for systematically so far compared to a 15 point something, I don't actually remember. So maybe it was two, maybe it was something else. For something we've been looking on for decades is pretty good and probably a good indication that we want to pay more attention to it in general, right? Okay, moving on. Can we find new stuff? This is all the stuff in ClinVar. That's what people know. Maybe we help them annotate this. That's good, but can we find new stuff? So that we did what's called PheWAS, Phenol-Wide Association Analysis, and we used the Penn Medicine Biobank for that. So this is an old slide. The number of sequence participants is well over 44 now. But there's a breakdown between different ancestries here, and we focused on European and African just because of power in terms of the number of samples, and of course we had to exclude related individuals, etc. And then we did PheWAS, focusing specifically on breast cancer because here there is a big problem of various of unknown significance. So there is genetic inheritance that we still are unable to explain. And also we have collaborators who are experts at this and have additional unique data sets for that and expertise. So we did two things. We got a list of genes they are interested in, and we also did this PheWAS to find new hits. And so one of those hits that I'm going to show here is EPN3. Very strong hit in breast cancer that you see here. And this is like the FDR and Bonferroni thresholds on this. That was not on the list that we got, but then we looked at literature and we find multiple papers showing clear association between breast cancer and EPN3. So that was one example of new things that we found. And the other one was in a variant in a gene or in multiple genes that we got, whether it was a PheWAS or not, that we decided to go ahead and validate. Remember, the last part for us is validation. So for that, we used a dual luciferase assay that we developed in the lab for this task. So we can introduce the 5' UTR, in this case of EPN3, with or without the genetic variant. And you can see here that a single mutation of the 5' UTR that hits that G-quad has a very strong effect between the wild type and mutation in 3 different cell lines. This was all done by Danielle Gutman, who was a postdoc at the lab. And we did the same thing for variant in MSH6 that was on the list of genes that we got associated with breast cancer, Lynch syndrome, and others. And the interesting part of this is a variant that actually introduces, doesn't break, okay, but introduces a G-quad and huge effect on downstream translation from a single mutation in our assays. All right. Now, this shows you function in terms of the phenotype that we can measure accurately with our assay. But notice that I sort of dodged the bullet of, do we actually have structures here, right? Which is this G-quad wars that I mentioned in the beginning, right? So do they actually form structures? So for that, we turn to Dan Dominguez's lab, the same guy who I mentioned from the bioRxiv preprint. And they tested those genetic variants that I just showed you for G-quad formation and disruption. And they see very clear indication with this circular dichroism experiments at different temperatures and with lithium or potassium for forming, if you remember from the start of my talk here, the formation, this characteristic profile, and disruption of the changes between mutant and wild type, both for the MSH6 and the PN3, exactly like was predicted by the model. Now, of course, this is a low throughput. That's one limitation. It's also not in vivo. So I still haven't proven to you that this structure forms in vivo. What we have shown is that those models seem to be very good at predicting the effects, and they connect to phenotypes, and they connect to structure phenotypes in vitro assays. And yeah, so that's where we are with this. And of course, this is still low throughput, but maybe your variant of interest is different and it's not one on our lists. So we also developed a web tool. You can plug in any sequence of interest and do those predictions. And I think the released version and that response, I think, Julian, you asked me in Switzerland. So I think we pre-computed Nomad and everything there is already there. So in the version that we will release. So I think that addresses that good suggestions that you made. Okay, so to summarize, I showed you GFormer, this language model based transformer for get insights into G4 subtypes, length effect, experimental biases that we discussed, flanking regions importance and context dependency, and RBP binding candidate selection for functional testing. I also showed you how we can connect it with patients' genome sequencing to find disease variant association, either in known or unknown with FIWAS genes. And I also showed you experimental validation of variant effect on function and structure that shows strong and context-dependent effects. All right. Future directions that worth exploring that we're very interested in actively working towards is predicting those RT stop scores, right? So I just showed you the preprint from Dan Dominguez's lab showing all those measurements for all those genetic variants. So you'd be like, well, you have all those measurements. Would you be able, and they're quantitative. They're not just like, oh, it forms not or not. They like give you a value that represents a strength. So can you predict this? And so the short answer is yes. These are like results from like the last week or so. So this is our model, the transformer model. And this is test data on that specific data set that I showed you before. And in case you're wondering, well, might be a very easy problem. This is what we get when we do a XGBoost decision tree. So yes, you can predict things. And if you're wondering what happens if you do a CNN, that's what we got. Okay. And we haven't looked, these are all very new results, fresh off, not the press, because it's not published, of course. But what we think is happening here, as an educated guess, is that really the decision tree is not very good at finding structures or detecting them. And then the CNN is also not as good, but the transformer model is much better equipped at those. And I think, running interpretation methods like Julien's recent paper would be a good way to investigate this further. So that's one thing we're very interested. Then the interconnection between RG4s and RNA binding proteins, we're very interested in that. The effect on phenotypes that are associated, expression, translation, splicing, etc. And then if you can figure all those things out, we're also very interested in RNA modulation. So how can we modulate this? And that would lead me to the second short part of the talk. But before I do this, I want to acknowledge all the great people that did the work. This was all led by a very talented computer science PhD student in the lab, Farika Zhang. The experiments were done by Danielle, who was a postdoc, and there were and other contributions from Nate and some and our collaborators, Kate, who is the breast cancer expert, and Dan Dominguez and Brian, who are collaborators on structure. Okay, so I talked about modulation at the end as something of interest. So for that, I'll switch to sort of the shorter part of this talk and talk about the different mechanism of RNA affecting RNA processing, and that is one for upstream open reading frames. So just to orient everybody, if this is the mRNA transcript here, and this is the 5' UTR, we can have various versions of start and stop codons here that can potentially be contained completely within the 5' UTR or overlapping it or introducing just an alternative start as well. All of those versions can and do occur. And of course, they have different effects. When you think of the ribosome machine, the 40S, with a small subunit scanning along and coming and starting to scan here, recruits the 60S, then they can do different things. They can get here or maybe not recruit or deassociate, etc. And all of those different variations of what can happen because of those UTRs affect their downstream effect on their effect on the downstream gene. So ribosome dissociation, ribosome stalling, NMD induction, these are all sort of downstream effects of this that can happen. Or you could also potentially have even encoded peptides, micropeptide. And you can have reinitiation or bypassing of this. It's what's called leaky scanning. Okay. And that will, of course, have an effect of increasing or decreasing the downstream gene translation. So again, we went about assessing using population genetic tools and statistics to try and assess whether UTRs are indeed regulatory elements under negative selection. And we published this paper in 2021. Notice this is the same author, a very talented MD PhD in the lab that published those back to back, almost not back to back, but like 1.5 year apart Nature Comp papers, one on the UTRs and on G-Quad. Here he also did all the experimental assays to prove their function. Okay, so that paper led us down the route, and again, David was the one leading this originally, for can we utilize this being an MD PhD, right, with an eye on therapy? So can we use this to potentially target for therapy? And we focused on the BMP pathway. I'm showing here a schematic of it on the left, and the list of genes that are associated with it, 8 genes, 3 different diseases. And we focused on a disease called pulmonary artery hypertension and the key gene for that, which is BMPR2 as a starting point. And so if you look at the 5' UTR of BMPR2, it has a lot of UTRs that you can identify them, and you can see a lot of them, and they're also overlapping, and etc., which is a very common phenomenon, which of course makes it a harder problem. But PAH is a very motivating disease to look at. It doesn't currently have a cure. It affects about 1 in 20, mostly women. Median survival rate is less than 5 years with solution of lung transplant and increased risk after COVID-19 infection. And so the industry have been looking for a cure for this and for a lot of time. And that's again associated with insufficient production of the BMPR2 gene. And so we wondered whether we can use our computational pipelines to identify UTRs that we might be able to target. And we tried a few that came up as potential candidates. This is the results of increasing translation, and each one of them is trying to target a different area. The ones in gray are basically like wild type. The interesting ones are the purple and mostly the tower in the center in yellow, which represents about 200%. So a twofold increase in translation, which is exactly what you would want if you lost one copy in this disease. We also measured the effect with luciferase assays when we treated this or target this with ASOs, and we saw a similar trend. We went to patient cells from a database that happened to be conveniently at Penn, so that made the process much easier. We got access to patients' samples. This is one of them. Again, we see a very nice increase in protein levels for BMPR2. And then we injected ASOs to live mice, and again, we saw a very nice increase in BMPR2 levels in pulmonary artery, not in other tissues. So just to summarize this brief part, we can combine experimental and predicted UTRs through our pipeline to identify candidates for functional testing, and they can be strong modulators of translation in general, and ASO targeting can modulate translation. I would just point out that I like to quote my friend who says it's easy to cure a mouse. So, we still have work to do to get it to patients. So with that, I want to acknowledge the people who did a lot of the work. David is at the bottom left here. He started this, and then Danielle, the postdoc who worked on this together with Nick Hand and other people in the lab. Isaac was a tech who helped, and staff development, and a lot of people that you see here at the bottom that helped us get through all the IRBs and experiments and access, etc. And of course, all the funding for this. So I'll summarize this part and connect it to the previous part with this slide, trying to explain, what did we learn today? And I would argue that what we learned is that dedicated predictive models for RNA processing combined with genetics can really gives you a push towards target identification, the what and the where, which is what we went after, plus mechanistic insights, right? Especially if you pay attention to what the model is telling you. And with that, in the talk at Switzerland, actually, because I talked to some of the students there, I added some visual commentaries that appeared to have been well received. So I wanted to sort of reiterate them. Maybe we have more audience here, and I'm guessing there's a lot of students, so I will repeat that in the hope that it makes more people think. I mean, it's good points to think about, also for us as PIs. And so these are my parting visual comments. The first one is you really want to think carefully. Do you want to get into model and data size competition? The GIF here is a spoof on Star Wars, and he's commenting that his Schwartz, which is the force, is his force is at least as big as his. So they're dueling like in the classical Star Wars scene. And the point is, if you think of model and data sizes and what you're developing, say, in academic environment, you're probably going to lose out, I would argue, against, the recent Alpha Genome or Illumina, or, I don't know, people who process all genomes of all living things or whatever, right? So, you know, think of whether you want to get into that, or maybe you want to. I think there's a lot of that now, and there's been a lot of those. We have these models and we've processed that data, but then the question is, what do you do with them and what do you achieve with them? And that leads me to my second comment, This is the Turkish ice cream seller. If you haven't seen the full clips, I encourage you to see them. They're very fun to watch. It's just a small illustration of this. But really, the point to make here is, what do you want to do, right? So if you think of getting, eating the ice cream as maybe, finding the stuff that you want to find, you can do this in an engaging way, maybe through very uh, sophisticated or maybe very impressive models. Do you want to get the ice cream? Do you want to play with the ice cream? Do you want to just sell the ice cream? Which, all of those are legitimate, and depends on what you want to do with your career, with your life. But these are good things to think about. To be fair to the ice cream seller, maybe his ice cream is amazing, and if you're focused on selling, I think he became a celebrity and probably got a lot of people to come to his stand. So again, what do you want to do if you think of it in terms of models, results, and things like that? And I think this is something that we think about as PIs, we constantly think about. But I think it's also good for the younger audience that think moving forward, what kind of science, what do they want to focus on, right? Because, the fact of the matter is, we have limited time. And so everything you choose to focus on means also that you're choosing not to do other things, right? So I'd like to say, we only get that many rides around the sun at the end of you know, so you want to think how you want to spend that time doing what. And with that, I'll thank you. I'll thank the lab who's doing all this amazing work and all the finding agencies that have helped us get everything done. And thank you.